So today I'll be talking about a lot of the work that I'm doing for my dissertation project. This is characterizing the glycosylation systems in the Mycoplasma species. Most of my work focuses on M. genitalium. However, I will mention some synth work in a little bit. Just for a quick review for those of you who aren't mycoplasmologists, or in the event there are any. M. genitalium is a bacterial STI. It is acquiring antibiotic resistance at a pretty alarming rate. Obviously, in addition to the genus's inherent resistance to anything that targets cell wall biosynthesis on the account of it not being there, it's also picking up resistance to the one or antibiotic classes that are uh, effective against this species. Uh, I include a very important caveat that this is the smallest genome of any naturally occurring free living organism. Um, so we kind of use, or you can kind of look at it. Or sorry, I should say Syn 3.0 on this title here. But we try to look, or the way that I've been looking at this, using these two organisms as a model, is that M. genitalium represents the natural, or as we know of right now, the smallest naturally occurring genome, or instance of extreme natural genome reduction, whereas Synth3b is artificially taken place. Only pain with working with M. genitalium, as you can see here, is it has a significantly longer reproduction time. It's a pretty fastidious organism to work with, and also M. genitalium is pathogenic, whereas I guess in most cases Synth3 is not. Uh, So glycosylation in a typical bacterial system takes place by an enzyme called a glycosyl transferase, which will donate a UDP sugar to a target protein. However, we have noted through carbon-13 experiments that most Mycoplasma species aren't using that system to glycosylate their surface proteolipids. To look through these or to look at glycosylation in mycoplasmas, we've been using a kind of a screen where we use a chemical species called periodic acid, which can specifically cleave a functional group motif called a vicinal diol, just two hydroxyl groups next to each other that you find in the ring structure of a carbohydrate. It undergoes a chemical reaction called the Malaprade reaction, which will cleave the bonds between the ring and yield either a aldehyde or ketone functional group, which a specific dye called ProQ Emerald 300 will then bind to and fluoresce. Of course, one thing we quickly were realizing when we work with M. mycoides, especially, or say specifically the GM12 wild type or Synth1, is if you look through some of the old Mycoplasma mycoides literature, it produces a very substantial amount of a 1,4-galactan, which, as you can see, has quite a few vicinal, or rather a vicinal hydroxyl group on it. So we kind of realized pretty early on that we need to treat these samples with galactosidase and do dialysis with them. So over here I have an M. genitalium gel where we loaded one sample with, or say we loaded two gels with the same quantity and quality of material. One of them just, you can use any dye for protein composition, and another one in tandem with that being that Emerald 300 glycostain. So we've noticed two types of glycosylation systems in this genus. As you can see here in band A, as just a kind of arbitrarily chosen example, and they have like a roughly like 1-to-1 signal ratio. We've called this one. This one has been characterized by James Dobenspeck earlier in the 2010s in M. pulmonis and pneumoniae and arthritis to be a hexoscillation system, which I'll get into in just the next slide, actually. There's also another one you can see here is exemplified in band B, where the glycostain is significantly brighter than the Kumasi stain right here on the right. And you can see there's these certain regions here when you quantitate the pixel intensity. And so we've hypothesized that these two glycosylation systems have different compositions and structures to them, and also have a different physiological role. So looking at band A, or that band A that I showed right there, this is that type 1 hexoscillation system that was taking place. As I mentioned with our carbon-13 labeled experiments, it's been determined that this donor glycan comes from the host or the environment, or an oligosaccharide from the host or environment, where it looks like it's being, or the hexose is being cleaved through the glycosidic bond being removed. Um, and this is a stochastic process that can occur to O and N linked amino acids, most notably glutamine, which is a pretty rare glycosylation event to see in bacteria. We
Hmm.
believe that this is an essential process for survival because we haven't been able to knock this out in any transposon library. Um, and we think, or the current reason that we, or I guess our model for why that might be is it might be, or we think that it could be protecting from protease digestion. And so ultimately, we think that this is coming just from a kind of scavenging of either like, your host or environmental polysaccharide, as I mentioned. The type 2 system, as I mentioned, is a little bit unknown. If we, I guess I'll jump back actually quickly. If you look at this GC chromatogram from that first band, you see that this is a very, there's lots of instances of glucose and mannose taking place. Whereas this type 2 system has much lower glucose and mannose levels and a significantly higher galactose within the excised band. We think that it's a oligosaccharide, as you saw from the intensity of that signal relative to, say, those hexoscillation system or hexoscillation bands. And we think that a novel enzyme is doing this. Uh, there's only one glycosyl transferase that's like canonically can be found through a homology search, and it's essential. And one thing to keep in mind is that both of these glycosylation systems increase antigenic variation across a population of mycoplasma cells. So, each one's going to be immunologically distinct from its neighbor. It's just stochastically adding on these hexoses and then also using this type 2 system. We believe that it's non-essential, or at least that's what we're kind of hoping for. And it's involved in immune system evasion, or we think that, as I mentioned, due to this antigenic variation, there's a immune system evasion aspect to it. For where and hopefully Synth 3 will be able to be helpful to us. Um, I will emphasize that this is, very preliminary data. I'm actually, once this workshop ends, I'll probably be running to try to do some repeats on this and clean it up a bit more. Um, so currently in progress, we're doing a protocol actually published by Kim Weiss, using Triton X114 extraction to purify out these proteolipids. And as I mentioned, this is very preliminary data, but we're hoping that the Synth 3 doesn't have this type 2 system. Uh, Obviously, I need to get more repeats of this. I want to clean it up, and then we'll try to characterize it further. Um, Another thing that the our collaborators at the JCVI have given us is also a M-genitalium transposon library where 97 of the 482 genes have been knocked out. So we're hoping that both Syn3B and these M-genitalium transposon mutants will help us to elucidate what genes are involved in the type 2 system. Oops. And lastly, I think a lot of, or I say like I guess the bulk of my long-term work will be doing Fourier transform ion cyclotron resonance mass spec on purified glycoproteins to determine the sites of glycosylation as well as the glycan structure, which will basically just be involved in purifying out our glycoproteins, screening a lower resolution mass spec to confirm our purification techniques and also see what proteins are there, and then sending these off to one of the dozen or so, or one of the dozen or so FTICR facilities, which will give us incredibly high resolution mass spectra to work with. With that, I'd like to thank my lab, especially the UAB Diagnostic Mycoplasma Lab. They're very helpful, especially for working with, as I mentioned, M. genitalium is kind of a painful organism. My co-mentor, Kevin Dibvig, are collaborators at the JCVI, my committee, and UAB Pediatrics for funding this research. And with that, I'd be delighted to take any questions that you have.
Anyone? I heard a voice. John, in a broader sense, glycosylation is a universal phenomenon in, I guess, all life forms, viruses, bacteria, eukaryotes, archaea. So what is this suggesting about glycosylation in bacteria in general? Is this something that the mycoplasmas have gotten rid of most of the systems, or is this where do you see this in an evolutionary light as you think about your experiments?
Well, I'm sure an obvious, I guess, motif that many of us studying, especially the synth organism, um, is that if this organism can tolerate losing something, it will, and then it much as a life, as a parasitic lifestyle, prefers to scavenge things. And so I think that at least from what we understand it so far about this process, it looks like it's a rather elegant way to protect itself and hope, like I said, hopefully if we're correct, increase its antigenic variation. But I think that it really demonstrates that these organisms have I view them much more as a well, I say at least for genitalium, maybe like a very like streamlined or optimized pathogen more than I guess viewing it kind of like simply as a you know I guess view them as a simple organism rather than a streamlined organism.
Kevin Dibving and I have long had discussions. His view that glycosylation in capsule is how mycoplasmas keep from fusing just by a process of membrane fusion, and that may be why the glycosylase activity is essential, because while I can see that glycosylation may be necessary for life in a goat or a human urogenital tract, it's probably not necessary for life in SP4,
Definitely not.
except for maybe that keeping all the cells in your culture from fusing.
Yeah, I think that'd be also another interesting one to look at. Yeah, I'm really hoping that we can get some good insight from what uh, if we find some transposon knockouts, what their putative functions are. Because yeah, that also sounds like, or yeah, that's one thing that's definitely always boggling my mind thinking about is the aspect of everything still taking place in SB4.
John, part of the problem, this is James Dobbinstead, by the way. Part of the problem is that it's not really, it's almost like the molecules and the rest of the bacteria evolved along two separate lines, and as far as glycosylation goes. The hexosylation system, which is shared by all molecules that we have looked at, including SYN3, is a unique system. It takes oligosaccharides from the host or the media, and before the oligosaccharide ever enters the cell, the bacterial cell, the enzyme grabs a hold of it, cleaves it, uses that energy to stick a hexose on the protein on the outside of the bacterial cell. This is a unique system. And the other system we think would be unique to molecules as well. So, and that plays into all the problems we've had with homology and, just predicting what protein does what. Mycoplasmas have found a different way to do things. They still do the same things, just a different way to do it.
Why do you think they evolved that, James or John?
It's a good question. I'd love to have an answer. And that's why I found some of these talks so interesting, especially the directed evolution talks from last month. But I know a lot of the people in this group didn't come from the mycoplasmology field, right? So maybe they don't have quite as good a grasp on the idea that mycoplasmas are this very unique, and I include all molecules and spiroplasmas, but that's a discussion for another day. But this genus of bacteria are truly unique. You're going to find there's stuff that we haven't even dreamed of yet here in these organisms. Synthetic pathways that are completely different, go completely against the dogma of regular bacteriology. Like I said, we've already identified 2 systems that way. We have found ABC glycosyltransferases, which are ABC transporters. If you do homology, they'll come out as ABC transporter permeases or some variation of such. But their true function is they synthesize capsule and excrete it across the membrane. We've found 2 examples of this already, in Mycoplasma and in M. genitale. And directed evolution talks from last Monday, there was an ABC transfer permease that was knocked out in Cent3O. I'm betting dollars to donuts that is a ABC glycosyltransferase that's no longer it would be essential in a host, but not essential in vitro. So,
could that be, if you had the gene for that, could you express that in, if you had the codon, the standard genetic code version of the gene, could you express that and test that?
I don't see why not, but it would probably be a pair. So it's probably and then it would probably be an operon actually. I looked at the gene structure and Mycoides genome yet. But you would probably see an operon or 2 genes that are really close together, because most of those exist as a heterodimeric pair.
That's worth looking into. That's a potentially interesting project. We need to move on.
Can I ask a naive question?
Sorry for the time, John.
No. Anyone else?
Could I ask a short question real quick?
Make it so.
Yeah. Maybe this is a bit naive, but how much is known about how much the glycosylation on the surface contributes to the mechanical stability of the cell to physical perturbations like osmotic shock?
We don't know a whole lot, mainly because I'm not really sure how to ask that question. If you can think of a real good way to ask it, we'll be happy to ask it and try to answer it. But I think it's quite possible.
Yeah, I guess how I'd answer that is that we're still, I feel like it's such like trying to understand like on the basal level how everything's going on, or like what's going on, that I really would love to answer that, but I feel like we need to get to the other problems first, or get through these other problems before we get there. But I do think that's a fascinating question.
Thank you very much.
Thank you.